Thank you very much for the invitation to come to give this talk. There's a lot to squeeze into this half hour, but I'm going to try my best. So my talk's on epigenetics, imprinting and assisted reproduction. I'm from the University of Leeds in the Division of Reproduction and Early Development. I'm going to cover an introduction to epigenetics, epigenetic reprogramming in the gametes and embryos, current knowledge of imprinting in human embryos, about ART-induced epigenetic disorders, assisted reproductive technology techniques associated with epigenetic disruption. I'll talk about infertility and epigenetics and epigenetic changes with in normal ART offspring. Can you hear me at the back? Everyone okay? So, an introduction to epigenetics. Okay, thank you. Cheers. So, in biology, the term epigenetics refers to changes in phenotype or appearance or gene expression caused by mechanisms other than changes in the underlying DNA sequence. And we can think of it as another higher level or layer of information that's additional to that presented in a gene sequence. For normal development, the pre-implantation embryo requires epigenetic information inherited from the sperm and the oocyte, and without that information, the embryo is non-viable. And there's significant reprogramming of epigenetic information during gametogenesis and pre-implantation development, exactly at the time that we're performing assisted reproduction. Accordingly, assisted reproduction can affect epigenetic reprogramming and causing disease, and infertility itself is associated with epigenetic errors. So, epigenetic marks, these are molecular modifications that regulate gene expression and genome function, and they can lead to heritable changes in gene expression without changes in DNA sequence. And the major epigenetic modifications are histone proteins and the DNA methylation. These can affect gene expression, and there's differential marking of the maternal genome and the paternal genome. And what's important is that the early pre-implantation embryo can recognize the differences. So just to describe epigenetic marks in more detail, if we take a chromosome and we unwind the DNA as much as we can, right down to the level of the base pairing of the DNA, then one epigenetic mark is DNA methylation. And this is methylation of certain bases in the DNA strand. We also have histone modifications, and those can be methylation, for example, or acetylation of the histone tails. And that both DNA methylation and histone modification affect the chromatin structure. They can either cause an opening of the structure or a closing of the chromatin structure, and that's important because it can regulate gene expression. When I'm talking about DNA methylation, I'm referring to a covalent methylation of DNA, and that's a methyl group being added to a position at C5 of the pyrimidine ring of cytosines. That's often within the context of a CpG dinucleotide, so it's a modification of a base rather than a change of sequence. So in a sequence context, it's just a marking, a modification of cytosine, for example, rather than the change of sequence. And histone modifications, here we have specific amino acids in histones can be modified post-translationally. The modifications include methyl groups, phosphate groups, acetyl groups, and ubiquitin groups. And histone modifications modulate the chromatin state and gene expression. With respect to nomenclature, H3K4ME3 refers to a trimethylation of the fourth lysine on histone H3. So, epigenetic programming in gametes and embryos. Epigenetic reprogramming is global remodeling of epigenetic marks associated with major transitions of cellular identity. In mammals, the term usually refers to the remodeling of epigenetic marks during germ cell specification and following fertilization in the early embryo. And what is important to remember is that epigenetic marks can be erased, they can be established, or they can be maintained. So in this review by Morgan in 2005, describes nicely the epigenetic reprogramming cycle. So in primordial germ cells, epigenetic information is erased. And the genomes of the gametes are then de novo methylated and acquire methylation and acquire imprints. That's called establishment. And then fertilization signals significant reprogramming. The paternal genome is actively demethylated, and then the protamines in sperm chromatin are replaced with histones, and the histones bring with them a series of epigenetic marks. Embryonic DNA is passively demethylated during pre-implantation development in a process called erasure, but the imprinted genes step outside this process and maintain their methylation through this reprogramming phase. By the blastocyst stage, we have a differential marking of the inner cell mass and the trophectoderm with heavier methylation in the inner cell mass. So this illustration shows all those processes just in an image. We have developmental time from left to right here, and we have a degree of methylation from low to high. So if we start with primordial germ cells, here on the left, we have gametogenesis, we have fertilization, pre-implantation development. Here's the blastocyst, and here's the conceptus in utero. We have erasure of epigenetic information from primordial germ cells, establishment during gametogenesis in the male germline and in the female germline. And at fertilization here, we have two very specialized cell types. We have the sperm and the oocyte that have a very high degree of epigenetic information associated with them. So at fertilization, we then have stripping away of methylation actively from the paternal genome and passively from the maternal genome. And then by the morula stage, we have a low level of methylation in the pre-implantation embryo. And then that increases with increasing cellular differentiation. So in the zygote, the reprogramming is very nicely illustrated by this slide. We have example of mouse zygotes, and we're looking at active demethylation of the male pronucleus in the mouse zygote. So we have DNA staining on the right-hand column, and then we have methylation staining in the left-hand column. So 3 hours after copulation, both pronuclei are methylated. 8 hours after copulation, we have partial demethylation of the male pronucleus. And 10 hours after copulation, we have a complete demethylation of the male pronucleus. And by the 2-cell stage, both of those pronuclei are demethylated. So the reprogramming phases during in the zygote and early pre-implantation development are nicely illustrated in this slide, in this review by Albert and Peters, from the oocyte to the 8-cell embryo, and it superimposes all the epigenetic events alongside all the cellular and molecular events that we know well. And it just shows, for example, the active DNA demethylation of the paternal pronucleus and the acquisition of histone modifications on the paternal pronucleus as the protamines leave and histones arrive. In human pre-implantation development here in the mouse, this is staining for the 5-methylcytosine using an antibody. Here's the zygote on the left, and we have demethylation of the male pronucleus in the zygote, and then pre-implantation development from the 2-cell stage to the morula. We have genome-wide erasure of DNA methylation to result in a demethylated morula stage, but maintenance of methylation imprints, which I'll talk about later. And by the blastocyst stage, we have differential methylation between the inner cell mass and the trophectoderm, with the inner cell mass being more methylated. So this really nice striking image is of a 3D image of a mouse blastocyst stained for DNA methylation using an antibody against that methylcytosine. And you can see nicely the difference between the inner cell mass and the trophectoderm. So genomic imprinting. Genomic imprinting is a mode of gene expression that is highly regulated by epigenetic information. Genomic imprinting is when genes are expressed in a parent of origin specific manner, and imprinted genes are expressed from one parental allele, or either the maternal allele or the paternal allele. It depends on which gene you're looking at. And precise epigenetic mechanisms regulate imprinted gene expression. Computer predictions suggest that there's 200 imprinted genes in the human genome, but in the lab there's probably around 60 to 70 known imprinted genes. Many imprinted genes have a role in growth, including placental, embryonic, and postnatal growth. Examples include IGF-2, IGF-2 receptor, MEST, and H19. Diseases involving imprinted genes are due to no dosage or excess dosage of the genes, and the putative role of imprinted genes is in parent-offspring conflict model and possibly in restricting parthenogenesis. So a normal gene is expressed from both alleles, from the paternal allele and the maternal allele. An imprinted gene is different, it's expressed just from one allele. It has a signal to switch off one allele, and that allows the other allele to be expressed so that messenger RNA is monoallelic. In genomic imprinting, what happens is a mark is placed during gametogenesis. For example, here, a mark is placed on the maternal allele of an imprinted gene in the oocyte. This is methylation. This persists in the pre-implantation embryo. The sperm, in the same region in the sperm, didn't receive the imprint, and so in the pre-implantation embryo, this region has a differential methylation, and that leads to the monoallelic expression. So what do we know about genomic imprinting in human pre-implantation development? Well, understanding the epigenetic landscape of human embryos and gametes can help us understand the epigenetic signatures of normal development. And we can use the data to assess whether ART treatments are affecting the embryo. So research I've been involved with, way back in 1998, we looked at imprinting in human pre-implantation embryos. We designed an imprinting assay around a polymorphism in the SNRPN gene. This is an imprinted gene involved in Angelman syndrome. And we wanted to understand whether it was imprinted or not in the human pre-implantation embryo. So without going into too much detail, by screening parental samples and then screening the cDNA from 2 4-cell embryos, we were able to deduce that SNRPN is imprinted in as early as the 4-cell stage. And that's important because all the information is there in the pre-implantation embryo to dictate imprinted gene expression. We've expanded these studies more recently in Leeds to look at imprinting of the MEST gene. MEST is a gene that's involved in a fat metabolism and fat storage in cells, and it's an imprinted gene. And in these assays, we looked at a much larger cohort of human blastocysts. We've done everything on a single embryo level. We've taken the DNA from each embryo and we've genotyped it for a polymorphism in the MEST gene. That allows us to look at the transcripts and see if they're imprinted or not. So in each of these columns, we've either sequenced or we've done a restriction enzyme to deduce which are the informative embryos. And then we've looked at cDNA libraries generated from the same embryos. In these cases, using that technique, we're allowed to deduce whether the gene is imprinted or not in the pre-implantation embryo. And we observed 7 embryos with strict imprinting of MEST and 3 with relaxed imprinting. So we were intrigued by these cases of relaxed imprinting. We wanted to know what the cause was, what the consequences were. We extended the study to look at imprinting in a larger panel of cDNAs. Here we're looking at imprinting of MEST in a panel of cDNAs where we don't have genomic DNA for the embryos, we just have cDNA libraries from them. And we observed relaxed imprinting in 7 additional embryos. Where you see double banding, it means that there's a relaxed imprint. We did a similar study for H19, which is as yet unpublished, but we screened around 80 embryos individually. We identified informative embryos by screening the DNA, and then we looked at the cDNA, and without going into too much detail, we found H19 can be imprinted in the pre-implantation embryo, but there were cases where the imprints relaxed. A more extensive screen on cDNA panels again showed that H19 imprinting can be relaxed. So we really want to know know the consequence and the causes of these relaxed imprints in these embryos. Another way of looking at embryo epigenetic programming and the epigenetic health of an embryo is by methylation analysis. Here we're looking at pyrosequencing of single embryos. We're looking at KVDMR, RB1, and SNRPN, and GRB10, which are 4 imprinted genes. A true methylation imprint should be 50% methylated, and indeed there are imprints with 50% methylation in each embryo. Each of these samples is an embryo, and each of these dots represents a methylation at a particular CPG site within the differentially methylated region. But importantly, there's evidence for variable methylation between blastocysts. SNRPN was very variable, and there was variability in methylation between embryos for KVDMR. So I've introduced epigenetics. I've talked about research that we're doing in Leeds, but I think this is the most interesting part of the talk is about art-induced epigenetic disorders. So embryos or gametes that are cultured away from the reproductive tract experience physiological stresses, and ART is performed, as we've shown, exactly when dynamic changes in epigenetic information occur. So those epigenetic mechanisms are susceptible to disruption by certain environmental factors or ART techniques. And there's been a number of epigenetic diseases associated with ART. Infertility is also associated with epigenetic defects. The strongest association between ART and epigenetic disorders is for Beckwith-Wiedemann syndrome, Angelman syndrome, and now possibly Silver-Russell syndrome. These are all disorders of genomic imprinting. In 2002, 2003, there were a number of papers that described disruption of epigenetic processes by ART. For Beckwith-Wiedemann syndrome, and the link to ART, the data was that 6 of 149 children in the Beckwith-Wiedemann syndrome registry were born after ART. 3 were ICSI, 3 were IVF, and this was compared to 1% in the general population. A subsequent paper suggested an increased frequency of use of ART in children with Beckwith-Wiedemann syndrome. And what's interesting in these cases, in these rather unfortunate cases, is that there's a methylation defect of KVDMR1, so hypomethylation of a region of chromosome 11 that affects expression of genes in the neighboring region. To summarize the information so far, it's best to go to this review by Aderma and Seegers in 2010. This summarizes all the papers that have shown a link between assisted reproduction and Beckwith-Wiedemann syndrome. It describes the technique used, and the important thing I want to say is that nearly all cases of ART-related Beckwith-Wiedemann syndrome have a demethylation of maternal KVDMR1. Angelman syndrome has been linked to assisted reproduction. Angelman syndrome is disruption of genes on chromosome 15q11 to 13. 2 Angelman syndrome patients were found with defects at an imprinting control region, and these were conceived by ICSI, suggesting a link between ICSI and Angelman syndrome. And then another case after ICSI was found. Again, the review by Aderma and Seegers is the best place to go for the most recent information. And the important thing is, in a lot of these cases, that there's a loss of maternal methylation at an imprinting control region. So what I'm trying to convey is rather than a genetic mutation, we have an epigenetic mutation, and that's possibly what ART is causing, some kind of epigenetic mutation. So Silver-Russell syndrome has been recorded. The first case was this 2007 paper, Silver-Russell syndrome in a girl born after IVF with partial hypermethylation at the DMR of PEG1 MEST. And if you look back in some of the other studies, there's actually SRS cases due to hypermethylation of H19, at least 3 examples there. So Silver-Russell syndrome will probably be studied highly in the next few years because of this paper in Human Reproduction. And this found a 10-fold increased frequency of Beckwith-Wiedemann syndrome and Silver-Russell syndrome with ART. There was aberrant DNA methylation patterns at multiple imprinted loci of both maternal and paternal germline DMRs. and there was a mosaic pattern associated with those epimutations, suggesting that the errors occurred after fertilization. So I'm just going to talk about some ART techniques associated with epigenetic disruption. So cell culture is strongly associated with causing defects in epigenetic programming. There's very much information from the mouse. There's so much data, I can't go through it, but just to summarize, there's multiple reports of culture-induced epigenetic effects on embryo and placenta with long-term developmental consequences. And this can cause disease, deformity, and behavioral changes, including obesity in mice after in vitro culture. And those are in fertile mice. The most striking example of the effect of cell culture is large offspring syndrome, and this is due to epigenetic defect to IGF2 caused by in vitro culture. Two very interesting papers in humans. These two papers from the same group, the Dumoulin and Nelson papers in human reproduction. Human pre-implantation embryos are exposed to either vitrolife or cook media, and the result on birth weight is quite striking. We can see a difference in birth weight between the two media types. So with vitrolife having a much higher birth weight. So that's most likely an epigenetic cause. A really good paper is also this Schwarzer et al. 2012 paper. This is a recent paper that's tested 13 of the commercially available media, human media, on mouse embryos. It's shown strikingly different effects on the blastocyst rate and fetal rate on the embryos, and also on the cell lineage composition of the blastocyst in a cell mass and trophectoderm, and also on gene expression, particularly of metabolic genes. Superovulation is believed to play a role in disrupting imprinting during ART. Many examples of the papers. Just an example is superovulation affecting KVDMR1 in human oocytes. And superovulation would be expected to be having an effect during establishment. Well, actually, it seems to be having an effect during maintenance of imprints. And this is probably due to some effect on some maternal effect genes. In vitro maturation and in vitro growth, again, have been shown to have disrupt imprinting. An example is this paper by Borgol, which is abnormal H19 methylation after in vitro maturation of human oocytes. And for superovulation and IVM and IVG, it's possible that they're causing, we're going against the natural process, which is stepwise growth-dependent process of imprint acquisition during the oocyte growth phase. And this has been shown nicely in mouse work. Some emerging stories are between epigenetics and embryo quality and developmental rest. This story, 2009, low-grade human ICSI embryos had more demethylation of histone modifications than their IVF counterparts. In this 2010 paper on global methylation study, the arrested or delayed human embryos had inappropriate DNA methylation and also aberrant chromatin. Another interesting paper is the developmental rate of embryos seems to affect epigenetic programming. So in this recent paper, the fastest developing embryos were worst with respect to controlling imprinting. And here we have the fast embryos with a loss of imprinting of H19. So infertility and epigenetics are linked. A number of recent studies have shown this, and this field is going, it's very busy at the moment. For example, we have effect on moderate or severe oligospermia affecting imprinted gene methylation at H19. Further examples of classes of male infertility with imprinting defects. And as you can see, I'm coming to the end of the talk by the way this blastocyst is hatching. And what we have is we observe epigenetic changes in normal ART offspring. In 2003, when the first cases of Angelman syndrome and Becker-Friedman syndrome after ART were spotted, Marr and co-authors wrote a paper about the iceberg scenario, suggesting that whilst imprinting disorders may be rare complications of ART, there may be more common epigenetic errors associated which may affect susceptibility to disease, including cancers in later life. That was taken on in 2009 again in another paper, and then this review in 2011. In the discussion, we have subtle effects of ART on epigenetic regulation with late onset implications, particularly cardiometabolic abnormalities, as well as subtle genome-wide changes in DNA methylation. And in that review, there's a very good table that shows you when you compare between different treatments, between in vitro and in vivo cohorts, that in all except one case, then there is a difference in either methylation or expression of imprinted genes. So just to summarize, I've covered a lot of ground, but it's important to introduce imprinting properly, because otherwise it just doesn't sink in. We found out that normal development requires proper epigenetic information, and that epigenetic information is reprogrammed exactly when ART is performed. Monoallelic expression of imprinted genes occurs in human pre-implantation embryos, and they also have the methylation imprints to afford that imprinting. ART can, in rare cases, cause epigenetic disease. Some aspects of ART are suboptimal for epigenetic processes. Infertility is associated with epigenetic errors, and ART may in fact exacerbate epigenetic errors in infertility. There are subtle epigenetic changes due to media that can affect birth weight and other factors, and accordingly we must be aware of the effects from subtle art-induced epigenetic changes in normal ART cohorts. So I think it's important in terms of research in the human is that we profile epigenetic landscape of human pre-implantation embryos and oocytes, and we do it properly and we do it robustly. And using that information, we can then check ART treatments to make sure they're optimal. Okay, so I just want to thank the sponsors, which is New Life Foundation and Medical Research Council. Thank you.